Assalamu alaikum wa rahmatullah. Today we will start our discussion about the female reproductive physiology after pregnancy. So in the last lecture we discussed the female reproductive system before pregnancy, and today we will look at the events that occur after fertilization of the ovum by the sperm. So the main objectives of this lecture is to describe fertilization and to recognize the development and function of placenta, also to recognize the function, the endocrine function of placenta, and to describe the physiological functions of the placenta-derived hormones, and then explain the mother's physiological changes that take place during pregnancy. Our discussion today will focus on the initial stages of ovum fertilization by the sperm. This is a video on YouTube. It's very nice. Please watch this video before continuing with this lecture. So as we discussed in the previous lectures about the ovulatory cycle, ovulation takes place in the ovaries when the primary oocyte enters the first stage of meiosis. It will give a secondary oocyte with 23 unpaired chromosomes. So then it will will initiate the second stage of meiosis, but it will not be completed until it is fertilized. So the ovary will expel the ovum while it is a secondary oocyte with 23 unpaired chromosomes into the abdominal cavity during ovulation process, which is around the 14th day of ovulatory cycle or female sexual cycle. Then the ovum at this stage will be surrounded by part of the granulosa layer, which is now called corona radiata, and then will enter the fallopian tube with the aid of the fabricated end of the fallopian tube and the cilia that is lining these ends, which move actually with the stimulation of estrogen released by the female ovaries. And then the ovum will be transported in the fallopian tube with the aid of the contractility of the fallopian tube, the contraction of the smooth muscle cells, and also with the aid of the cilia toward the ampulla. And now, after finding or being fertilized by the sperm, then the second stage of meiosis will be completed to complete maturation of the ovum, and then we will have again 23 chromosomes, okay, that are ready to combine with the genetic material, the 23 chromosomes from the male sperm. And normally the site of fertilization in the fallopian tube is the ampulla. And actually after ejaculation, sperms will reach the ampulla of oviduct of the fallopian tube within about 30 to 60 minutes with the aid of the prostaglandins that are released with the semen from the male and with oxytocin from the female. So it will reach the site of fertilization and fertilize the ovum. If we look at the chart on the left, you will see that in the cervical canal, after 3 minutes of ejaculation, there will be 3% of sperms that survived this stage. And in the uterus, only about 0.1% of the ejaculated sperm will succeed reaching the uterus. And the fertilization site, the upper third of oviduct, there will be only 1,000% of the sperms that will reach this site. And that's about 30 to 60 minutes after ejaculation. So in the figure on the bottom, you will see the egg, the fertilized egg, which will start the process of implantation in the uterus. And that actually happens after another journey of the fertilized ovum across or through the fallopian tube. Meanwhile, the zygote is dividing now, and we will look into these details in next slide. Now, after the sperm penetrates the corona radiata, as you can see here in this schematic, via membrane-bound enzymes present in the head of the sperm, and it will actually bind to receptors in the zona pellucida. Then binding to this receptor will start or trigger the acrosome reaction, which will release hydrolytic enzymes, okay, that will start digesting the zona pellucida, which is a layer covering the ovum. Okay, it comes after the zona, after the corona radiata, we will have the zona pellucida. Now, the acrosomal enzymes will digest the zona pellucida, as you can see here in this figure, and then will make a pathway into the plasma membrane of the ovum. Then the plasma membranes of both the sperm and the ovum will fuse together. Now, the nucleus of the sperm will actually enter the ovum cytoplasm. As you can see here, the sperm midpiece and tail will stay outside, and only the sperm nucleus will enter the ovum. Then it will stimulate calcium release from cortical granules in the ovum. And this process actually will inactivate the receptors, the sperm receptors, which will block any further penetration by other sperms. Okay, so this is why only one sperm can fertilize the ovum. The rest are going to be forbidden or going to be prevented from entering the ovum. So, and on the figure on the right, you will see the ovum, how the sperm nucleus will now start aligning to combine with the DNA or the genetic materials from the egg. And the figure on the bottom will actually is describing this process. We will have now a pronucleus for the male and a pronucleus for the female in figure D. So the male pronucleus will align with the female pronucleus, okay, with centrosomes on the two poles of the fertilized ovum. Then now the genetic material, the chromosomes, align in parallel to each other, the male and female. And at this stage, we can say that this is a complete union of the sperm and the egg genetic material. So after union of both male and female pronucleus, the 23 unpaired from the male and the 23 unpaired chromosomes from the female, then the fertilized ovum will contain 23 paired chromosomes, or the complete set, the complement of 46 chromosomes. And this is how the zygote will look just before the union of the male and female pronuclei in the fertilized zygote. So following fertilization, the zygote will undergo several mitotic divisions inside the zona pellucida, and the overall size will not actually change. After the first cleavage, there will be a 2-cell embryo. Each cell is called a blastomere, and the divisions will continue rapidly until the stage of 32-cell stage. After that, traveling of the fertilized ovum will take place across the fallopian tube, and it will finally reach the uterus and will implant into the lining of the uterus. In this schematic, you will see the timeline of fertilization and after fertilization stages from day 0 of fertilization until the completion of the fertilization, the zygote process. And then at day 1, the first cleavage take place when 2 cells are produced, then the 4-cell stage, and the 8-cell stage, which we call now morula, at day 4 and at day 5, at which the completed morula or the compacted morula will enter the uterus. And about the day 8 to 9, then there will be an implantation of the fertilized ovum, which is now a blastocyst. We will look at these stages in more details. So after 3 to 5 days from fertilization, the zygote will reach the uterine cavity. And the transport of the fertilized ovum or the zygote will be aided by fluid current, as well as the action of the cilia in the fallopian tube, with weak contractions of the fallopian tube by the semen and prostaglandins in the semen, as well as the action of estrogen. In the isthmus, there will be relaxation under the progesterone that will lead the entrance of or aid the entrance of the zygote into the uterus. But there will be a time of delay of the transport, which will allow cell division to take place sufficiently before entering the uterus. At this stage, a blastocyst that contains about 100 cells will enter the uterus. And as you can see here in this schematic, in B, the early cleavage, the 4-cell stage. In C, you will see the morula, which is about 8-cell stage, and the early blastocyst that will be implanted in the uterus. You can see a blastocyst cavity, and the late blastocyst, which is the exact implantation time. It will be composed of trophoblasts, okay, and will have an inner cell mass and trophoblast layer, and that's what we call late blastocyst that will be implanted into the lining of the uterus. So the trophoblastic cords from the blastocyst will implant and will start making the placenta. Blood capillaries will grow in these cords from the fertilized ovum, and after the 21st day after fertilization, blood will start to be pumped by the fetal heart into the capillaries. At the same time, maternal blood sinuses will develop around the trophoblastic cords from the implanted zygote, and more trophoblast projections will develop, which will form the placental villi. As you can see here, this is the stage of implantation. This is the uterine wall, the endometrium layer of the uterus, and here is the trophoblast, which is the surface layer of cells of the blastocyst, and actually the trophoblast will invade the lining of the endometrium and will form cords of trophoblastic cells. And in the figure on the right, you will see these cords growing, and the part of the maternal will be called decidua now. It was called endometrium, now it's called decidua. And you will see that amniotic cavity will be formed at this stage, as well as you will see the inner cell mass that will form the embryo. Placenta is the primary source of nutrients and gases for the developing fetus beginning of the 8th week of gestation. Before that, the developing ovum or the growing ovum will get nutrients in the fallopian tubes from secretions, the fallopian tube secretions that are full of nutrients with the aid of progesterone hormone, as well as in the uterus. Also, the uterine milk will also provide nutrients for the developing and dividing ovum, the uterine milk under also the actions of progesterone. And after implantation, also the blastocyst will get its nutrients from the endometrial lining that has stored a lot of glycogen, lipids, and a lot of nutrients by the actions again of the progesterone during the ovulation cycle. As we know, the developing or implanted ovum will still get these nutrients until the 8th week of gestation by these means. After that, placenta will start giving nutrients to the fetus, and actually it starts even earlier at the 16th week, 16th day after fertilization. But the main source of placenta will be around the 8th week of gestation. So in the developed or mature and developed placenta, the fetus's blood will flow through 2 umbilical arteries into capillaries of the villi. As you can see here, the placenta is composed of villi, the villus here, that comes from the fetus or the fetal vessels, and then we'll go back through a single umbilical vein into the fetus. So the umbilical arteries are not going to be oxygenated. They will get oxygenated in the placenta and go back through a single umbilical vein, okay, in the umbilical cord. That is going to be oxygenated from the placenta, and after being exchanged, the gases are going to be exchanged with the mother or maternal vessels in the placenta. At the same time, the mother's blood will flow from her uterine arteries into large maternal sinuses. Okay, so from the mother there will be sinuses, and from the fetus there will be villi. And the villi then, the sinuses will be surrounded by the villi, of course, to facilitate the exchange between the maternal sinuses and the fetal villi. And then the maternal vessels will go back through maternal uterine veins, of course, of the mother. So the exchange processes take place via diffusion, very similar to what happened in the pulmonary capillaries and also in any capillary beds in our bodies. So the main function of the placenta is to provide oxygen for respiration for the fetus, also to provide nutrients for the developing fetus in order to get the wanted or the needed required nutrients, amino acids, glucose, sugars, and also to provide excretory function for the fetus to excrete waste products, metabolized hormones, or metabolized metabolism products of many biochemical reactions in the fetus. Also to provide an endocrine function for both the fetus and for the female during pregnancy, and also it provides protection for the fetus. So these are the main functions of the placenta, and we will discuss in more details these functions. You can see on the right how the maternal blood and the fetal blood are related to each other. You can see that oxygen and nutrients will be diffused or transported from the maternal circulation into the fetal circulation, while CO2, carbon dioxide, and waste products will be eliminated or released into the maternal circulation, which will provide routes for eliminating these products. Of the important factors that facilitate the oxygen delivery from the maternal to fetal circulation is the difference in the partial pressure of oxygen concentration between the maternal and fetal blood. And although the difference is not very huge or very large, it's only about 20 millimeter mercury between the maternal, which is about 50 millimeter mercury, and the fetal, which is about 30 millimeter mercury. The difference is not large, but it is sufficient to facilitate the delivery of oxygen to the fetus, due to the other reasons that also facilitate the delivery of oxygen into the fetus, regardless of this very low difference. The high fetus hemoglobin in the fetal circulation, which actually has higher affinity for oxygen than the mother's hemoglobin. So fetal hemoglobin is very well known to have a dissociation curve for oxygen that is shifted to the left, as you can see here in the graph. The dissociation curve for oxygen for the fetal hemoglobin is shifted to the left. That means the fetal circulation has, or hemoglobin has, a higher tendency to bind and combine with oxygen than in the maternal hemoglobin. So even at the low gradient of oxygen between fetal and maternal circulation, the fetal hemoglobin can still carry 20 to 50% more oxygen than the maternal hemoglobin can do. Also, one of the other important factors facilitating delivery of oxygen from maternal to fetal tissues is the high cardiac output in the fetus, which is due to the combination of the cardiac output from both ventricles. That is due to the shunting processes that take place in the fetal circulation, and more importantly, the double bore effect that take place in the between fetal and maternal circulation. And this is explained by the Bohr effect, which is the high tendency of hemoglobin to combine with oxygen when there is a low CO2 level, and the opposite is true. So it will dissociate from oxygen when there is high CO2 level. In the fetal side, there will be high partial pressure of CO2, and that will dissociate from the fetal hemoglobin, leaving the fetal circulation, and that will make the fetal blood more alkaline, and there will be more tendency to combine with oxygen. In the mother's blood, taking carbon dioxide when it will be delivered or diffused into the mother's blood due to the concentration gradient, of course, this will leave the pH of the mother's blood more acidic, and that will induce dissociation of oxygen from the maternal hemoglobin due to the Bohr effect. So Bohr effects here occurred at the two maternal and at the fetal circulation, at the two circulation levels, and that's why we call it double Bohr effect. So even with the low gradient of oxygen between maternal and fetal circulation, the delivery will be enhanced to a high level due to this double Bohr effect. The double Bohr effect is explained further by using the oxygen dissociation curves, where important shifts take place in the placenta, and particularly in the maternal blood and in the fetal blood. So in the maternal blood, gain of CO2 from the fetal circulation will make the pH to fall, and then the curve will shift to the right. As you can see here, the red line, the red curve, the dissociation curve here is shifted to the right, making more additional release of oxygen from the maternal circulation. However, on the fetal side of the placenta, carbon dioxide is going to be lost because it will diffuse from the fetal into the maternal. So that will rise the pH in the fetal circulation, and this will cause the curve, the dissociation curve, to shift to the left. As you can see here, the green curve in the fetal will shift to the left, and that will allow more oxygen combination or association with fetal hemoglobin. So the double effects, the Bohr double effect here enhances more and more oxygen delivery from the maternal to the fetal circulation. Okay, so even at low oxygen gradient, this will enhance the oxygen delivery. Now let's talk about the nutrition of the fetus. The fetus will mainly have glucose as nutrition. In the trophoblastic cells, it will get glucose in the placental villi that is transported via carriers or transporters via facilitated diffusion through the transporters that are found in the trophoblasts. Fatty acids will diffuse due to the high solubility across the plasma membrane. However, it occurs more slowly than glucose, طبعا due to the availability of transporters بالتروفوبلاست cells. الافيلابيلتي لهم عالي جدا لدرجة انه بتكون الترانسبورت للجلوكوز اسرع من الترانسبورت للفاتي acids. The placenta actually have also active transporters for amino acids. With the fetal concentrations of amino acids will exceed the maternal levels. So that's why we have the active transporters for amino acids, not only the facilitator or facilitated diffusion process. Minerals or electrolytes will diffuse by diffusion from the maternal into the fetal blood with the bulk diffusion that take place, just like any capillary bulk diffusion, any capillary system bulk diffusion. The excretory products of the fetus will diffuse through the placental membrane into the maternal blood in order to be excreted with the waste products of the mother, of course, via the renal system, such as urea, uric acid, and creatinine. And higher concentration of excretory products in the fetal blood ensures that there will be continuous diffusion of these substances into the maternal blood. So due to the concentration difference, there will be a continuous diffusion of these waste products, which are products of protein metabolism or as the muscle metabolism. By this far, we finished our talk about the initial stages of fertilization and the initial stages of ovum development and fertilized zygote development and the placenta. In the next lecture, we will talk about the placenta as an endocrine organ, and we will continue our talk about the hormones and their contribution during pregnancy and gestation. Please post any questions you have so far on the e-learning forum so I can get back to you answering your questions, and good luck.